This is part 3 of Lecture 9 on acid-base balance. This information can be found in Chapter 26 of your textbook, pages 1013 to 1037. Regulation of sodium balance. Regulation of sodium balance is influenced by aldosterone and angiotensin II. Aldosterone plays the biggest role in regulation of sodium by the kidneys. And regardless of the aldosterone presence, 65% of sodium is reabsorbed in proximal tubules in the kidney, and 25% is reclaimed in the nephron loops of the kidney. Sodium is never secreted into the filtrate. When aldosterone concentrations are high, sodium is actively reabsorbed and water follows, so extracellular volume increases. When aldosterone concentrations are low, sodium is not actively reabsorbed and is lost to urine, along with increased loss of water. Renin-angiotensin-aldosterone mechanism is a main trigger for aldosterone release. Granular cells secrete renin in response to sympathetic nervous system stimulation, decreased sodium chloride concentration, decreased stretch of granular cells due to decreased blood pressure. Renin catalyzes production of angiotensin II and prompts aldosterone release from the adrenal cortex, which sit above the kidneys, and results in increased sodium reabsorption by kidney tubules. Aldosterone release is also triggered by elevated potassium levels in the extracellular fluid. Aldosterone brings about its effects slowly, in terms of days or hours. The influence of atrial natriuretic peptide, or ANP. The ANP is released by atrial cells in the heart in response to stretch caused by an increased blood pressure. The effects are decrease in blood pressure and blood volume, which inhibits antidiuretic hormone, renin, and aldosterone production, and increases the excretion of sodium and water. It also promotes vasodilation directly, and is also by decreasing the production of angiotensin II. In terms of the influence of other hormones, female sex hormones, such as estrogens, increase sodium chloride reabsorption just as aldosterone does, and leads to water retention during the menstrual cycles and pregnancy. Progesterone decreases sodium reabsorption and blocks aldosterone, and promotes sodium and water loss. And glucocorticoids increase sodium reabsorption and promote edema. Cardiovascular baroreceptors, or pressure receptors, alert the brain to increases in blood volume and pressure. The sympathetic nervous system impulses to the kidneys to decline, causing the afferent arterioles to dilate. Glomerular filtration rate, the filtration rate of the kidneys, increases. Increased sodium and water output is a result, with reduced blood volume and pressure. Regulation of potassium balance. Potassium affects the resting membrane potential. An increase in extracellular fluid potassium levels, known as hyperkalemia, will result in a decrease in resting membrane potential. This causes depolarization and is followed by reduced excitability. A decrease in extracellular potassium levels, known as hypokalemia, will result in hyperpolarization and non-responsiveness. Abnormal heart potassium concentration can lead to sudden death. Potassium is part of the body's buffer system. Hydrogen ions will shift in and out of the cells in opposite direction of potassium. To maintain a balance, extracellular fluid potassium levels rise with acidosis. Extracellular fluid potassium levels fall with alkalosis, and this can interfere with the activity of excitable cells. Extracellular fluid concentrations are the most important factors affecting potassium secretion. A high potassium diet will lead to an increase in potassium content of the extracellular fluid. Potassium entry into principal cells leads to increased potassium secretion, and a low potassium diet, or accelerated potassium loss, reduces its secretion and promotes its limited reabsorption. And this is the end of Part 3 of Lecture 9 on the acid-base balance.